I'm Professor Kushwa and welcome to another figure discussion video. In this chapter section, you're reading about transcription and the processing of one type of RNA called mRNA or messenger RNA. Now, before we look at the figure, let's discuss an analogy that may be helpful. My guess is that many of you have written long research papers for some of your classes, and you know that ideally writing the final version of your paper will not happen as a single event the night before it's due. Instead, you'll likely or hopefully write multiple drafts and edit them. You may add information or delete information before the paper is ready to be submitted, and the final version may look very different than the first draft. This editing process also happens with messenger RNA. In other words, the initial product of transcription, called the pre-mRNA, is modified substantially to produce the final product, which is referred to as the mature mRNA. This figure shows the three general steps associated with mRNA editing, which is also known as post-transcriptional modification. Again, post-transcriptional modification. First, a modified guanine nucleotide is added to the 5' end of the mRNA, and this is referred to as the 5' cap. Second, a poly-A tail, which consists of an average of approximately 200 or so consecutive adenine nucleotides, is added to the 3' end of the mRNA. Third, the intervening sequences, known as introns, are removed, and the expressed sequences, also known as exons, will be spliced or glued together. Now, at this point, you're likely asking yourself, okay, so what's the benefit of these modifications? Well, that's a really good question, and let's think about the answer. The 5' cap serves several functions. First, it protects the mRNA from being broken down or degraded by enzymes that are in cells called nucleases. Second, it participates in the transport process of the mRNA out of the nucleus through the nuclear pores. And third, it helps the ribosome attach to the mRNA for the beginning or the start of translation. The 3' poly-A tail also has several important functions. First, it participates in ending or terminating the process of transcription. Second, it also participates in transporting the mRNA out of the nucleus and into the cytoplasm. Third, it also helps protect the mRNA from degradation by enzymes. Most students understandably view splicing as an inherently inefficient process. I mean, why would you bother writing paragraphs of content for your paper if you knew they'd be deleted from the final version? Well, the most important reason this happens in transcription is that introns are present and they are necessary for splicing or the removal of themselves during the mRNA processing. They're also necessary for a process called alternative splicing, in which one gene can produce multiple mRNA versions that code for different proteins. Please note that I said one gene produces multiple mRNA versions, not that one gene produces multiple types of RNA. So for example, alternative splicing in this particular case might include the joining of exons 1, 2, and 3, while another splicing product might be the combination of exons 1, 3, and 4. Alternative splicing is an important reason why the number of structural genes actually identified in the human genome is so much lower than was initially expected. Remember, the initial estimates of the number of structural genes was approximately 1,000, and that number, as a result of further research, has been reduced to approximately 18,000. So I hope this explanation is helpful. And now let's take a look at several review questions. I'll give you 4 seconds to pause the video. Then you can read the questions and answer them. Once you're ready, restart the video and we'll review the correct answers together. So welcome back. Let's take a look at the questions and their correct answers. Question number 1, what process results in the production of mRNA? The answer is transcription. Question 2, distinguish between introns and exons. Which one of them is removed from the pre-mRNA or the immature mRNA during splicing? Introns are called intervening sequences, which means to come between. Exons are known as expressed sequences, meaning their information will be used in the process of translation to determine the primary structure of the protein. So exons are sequences that will be translated. Introns are sequences that will be spliced out of the mRNA during post-transcriptional modifications. Question 3, where in a eukaryotic cell does splicing occur? And the answer is in the nucleus. These modifications happen prior to the transport of the mature mRNA out of the nucleus and into the cytoplasm. Question number 4, what other modifications besides splicing are made to the pre-mRNA to produce the mature or final mRNA? I said the additions were a 5' cap and a 3' poly-A tail. Question number 5, what's the specific name of the chemical bond type that must be reestablished between the spliced segments, meaning the exons, to produce the mature mRNA? And what's the name of the associated chemical reaction? Remember, adjacent nucleotides are joined by a type of covalent bond called phosphodiester bonds. And those bonds are created by the process of dehydration synthesis or condensation. Now, if any of your answers were incorrect, I encourage you to re-watch the pertinent sections of the video to make sure you understand the basis for the correct answers. Thanks very much for watching this video.